This is part two of the viruses lectures. Um, really, this page just, it's got a lot of information on it. The most important thing is that viruses, well, let's start over. Living things, as we have learned, are made of cells, and inside those cells is either a nucleus or in prokaryotes, a nucleoid region that contains only DNA, just DNA. DNA, um, pieces of DNA contain the genes for living organisms. But viruses can either have DNA or RNA. Their genes can be encoded into DNA or RNA. And we're going to talk a whole lot more about that. In fact, I'm going to do a review of transcription and translation so that you can understand how viruses really are different from each other. But the one thing that I will say is that with RNA viruses like the COVID-19 coronavirus, they mutate at a greater rate, and so it's really hard. The flu virus, influenza, it's really hard to make vaccines for RNA viruses. HIV is an RNA virus, so these are viruses that RNA viruses tend to be a little more dangerous because they can mutate. They do often mutate. More than DNA viruses do, or more frequently. The way that a virus interacts with a cell, what you're looking at here is a human cell, and then the virion, or the virus particle. You can tell some things about this virus particle. You can tell that it's got, in its core, it's got nucleic acid. It doesn't say, you could Google it if you wanted to, because it tells you exactly what it is. It's the KSHV virion. It's enveloped. Most animal viruses are enveloped. The envelope is very similar to the cell membrane of the host cell. So the virus has the many structures. Well, first of all, let's go over the parts of the virus again. This is the nucleic acid. This is the protein capsid. This hexagon is the protein capsid. The envelope is the circular structure that has molecules that are attached to it, kind of like membrane proteins. They're not all proteins, but that's kind of how they are. The molecules that are found embedded within the plasma membrane, same type of molecules. So this oval green molecule is called a viral receptor. And what's going to happen is it is going to combine and have a shape that will match up with a receptor on the membrane of the host cell. And that's going to be the attachment phase. And by the way, the KSHV, that stands for Kaposi sarcoma virus. I may not have it. Anyway, back in the 80s, which many of you don't remember or weren't here for, the reason I can remember that particular virus is that back in the 80s, AIDS was it was a disease that was really, really scary. It still is, but not like in the 80s. In the 80s, it was a death sentence if you got HIV. So KSHV, the Kaposi sarcoma, it was like there were very dark, flat moles that would appear on the skin of an AIDS patient. And sometimes that's, you would look at someone and you could tell they had AIDS because they had that. That's just how I remember that one. But this isn't just an example of an enveloped animal virus. So moving on, here we have a bacteriophage T4, and this bacteriophage has a head and a tail. And The tail fibers actually have structures that look like little legs, and this is the one that we looked at earlier in our slides that attached to the E. coli bacterium. The head contains the DNA. The DNA is found in the head, and so this is just a complex shape, head and tail shape. The adenovirus has just the protein capsid. It has DNA as the nucleic acid, and it just has protein capsid made of capsomeres. The capsomeres are the little brown ovals. And then the darker brown kind of spikes, though they're not really spikes, but they're sticking out further with a rounded end. Those are the glycoproteins that help the adenovirus attach to the host cell. This is another image of an adenovirus, showing you a little bit better how it's more of a geometrical kind of shape. The word used is icosahedral. Icosahedral shape looks like this, or similar to it. HIV is an enveloped virus. Its nucleic acid is RNA, and it also contains the enzyme reverse transcriptase, which makes it really, really tricky to our immune system. And we're going to get to that when we learn about virus classification. But let's just look at the parts. We've got the RNA, which is the swirly yellow strands, and we've got the enzyme reverse transcriptase. All viruses don't have their own enzymes. Many viruses make use of the host cell enzymes, but this particular virus does. So the capsid of this virus is right here. You can just look at my pointer tracing out the capsid. That's the protein capsid, and then the envelope is the circular part, the spherical part. And the envelope contains matrix proteins, and it also contains what we're looking at here is a glycoprotein. And that is the part that will help in attachment to HIV attaches to T lymphocytes in humans. In other words, to our immune system cells called T cells, and shuts down our immune system. What we're looking at here are some images. This is the rabies virus photograph, and then also just an illustration showing you that it is enveloped, which we are not surprised by because animal viruses often are enveloped. You can see that the nucleic acid in the rabies virus is single-stranded RNA, but this virus does not contain any enzymes of its own, like any polymerase enzymes or transcriptase or anything like that. This is what a person with smallpox looks like when they're actively infected with smallpox. Thank goodness we've eradicated that disease, but this is an image of the smallpox virus. And its DNA is double-stranded, and it is an enveloped virus. So you've got two other examples of viruses that are enveloped that are animal viruses. That's very, very common. Okay, so I'm not going to go over this a whole lot. I'm just going to tell you that we are going to use the Baltimore classification system to classify viruses. It's the most commonly used classification system today, but this table shows you some differences between viruses. So viruses can have either RNA or DNA in their core. The RNA can be single-stranded or double-stranded. The DNA can be single-stranded or double-stranded. The chromosomes can be linear, like the chromosomes that are found in human cells. They're linear. We have 46 of them. And they can also be circular, like the chromosome found in bacteria. You can see here that many bacteriophages, which we know they're viruses that attack bacteria. They also have circular genomes instead of linear chromosomes. They have one long circular chromosome. And then sometimes the nucleic acid, the chromosomes or chromosome are segmented, and sometimes they are non-segmented. So there's just differences within the viruses. When we look at the shapes of the capsid, some viruses can be they say naked icosahedral. That's the shape that we saw in the adenovirus. Naked means it doesn't have an envelope, or it can be an enveloped icosahedral. Many examples over here on the right: enveloped helical and naked helical. The tobacco mosaic virus, we saw that was the first page that we looked at after the title page in our slides. And then some have complex shapes, like the head and tail structure of the bacteriophage. These are just different images. Here's our tobacco mosaic virus. This is an image of that. And it's inside these stick structures are spiral or helical viruses, kind of like this shape of a DNA molecule. Let's see. Okay, now the Baltimore classification. The Baltimore classification groups viruses into 7 groups, and it groups based on the characteristics of the nucleic acid of the virus and its mode of messenger RNA production. Now, the production of messenger RNA in cells is called transcription. So Baltimore classification looks at the nucleic acid itself, and it looks at how transcription occurs. And this is what we learned. This is a review. Transcription is the first part of the central dogma of life. We learned this back in Bio 111, that when DNA makes a copy of one of its strands into messenger RNA, that messenger RNA will code for a protein. And that part, when the messenger RNA codes for the protein, is translation. I have definitions here just to remind you, and I'm going to read them, even though I know you can read. Sometimes it's helpful to hear it. Transcription is the process where the double-stranded DNA inside of our cells unzips using the enzyme helicase, and one of the strands is used as a template or a blueprint to create a single-stranded messenger RNA molecule, and that occurs through the enzyme RNA polymerase. RNA polymerase comes along and adds nucleotides to the DNA template. So, for example, if this is your DNA template, it's double-stranded, so it's got two strands. The first one that I've highlighted is the template strand, and that one is TAC-CCC, and I've divided them into groups of 3 because, remember, 3 nucleotides will code for an amino acid in the protein. So for this piece of DNA, it's going to unzip, and the top portion is going to be used as the template strand, and it's going to produce a complementary RNA molecule. So if you go back over here, T is complementary to A. A in DNA is complementary to U in RNA, because RNA, remember, does not have the nucleotide thymine. It has uracil, so U. And then C's are all complementary to G's. Cytosines are complementary to guanines. And the AUG, if you look at a codon chart, AUG codes for the amino acid methionine. Remember, AUG we called it the start codon. And then GGG on the codon chart codes for the amino acid glycine. So what I'm going to do in the next recording is I'm going to go through each individual grouping of the Baltimore classification and compare it to the central dogma of life.